Welcome back, everybody. Today we're looking at Unit 3, which is half sampling design and the other half experimental design. And we start with a couple definitions. So population, census, and samples. A population is the entire group of individuals that we want information about. And in order to get information about a whole population, in order to be able to generalize to an entire population, we need to be very careful and make sure that we follow specific statistical principles that we learn about in Unit 3. A census is actually collecting information from every individual in a population, but we learn on this slide that we typically won't do that. And in fact, in this country, we only attempt to do that every 10 years. And there's several reasons for it. One is that it's very, very expensive, perhaps impossible. And there are certain types of surveys where we're using destructive methods to learn about individuals. Like, for example, if you're trying to learn the lifetime of a battery, the way that you do it is you drain the battery. Well, by the end of that process, that battery is useless. Right? So if you were to do that on all batteries in a population, you'd essentially destruct the entire population you're trying to learn about. So then we talk about a sample, and a sample is just a subset of the individuals in the population, and you're going to collect information from that sample. Hopefully that sample is representative of the population, and that'll allow you to make inference about the population as a whole. Now, a sample survey is one way to get information about a population, but if that population is very varied, you have to be very careful how you pick the individuals that are going to make up your sample. You also have to be careful how you phrase the questions to those individuals so that the results that you get are not biased. Now, in this unit, we learn both about really, really good ways about sampling and really, really bad ways of sampling. And we're going to start out with the bad ones. So the first one would be convenience sampling. And this happens when you approach individuals in the population that you're trying to learn about that are easy to reach. So for example, maybe we're interested in understanding the average number of books read by Americans in the last 12 months. And so we stand outside a library and ask people that question, how many books have you read in the last 12 months, as they walk by. Now this is super convenient because you're definitely going to have a bunch of people walking past the library in the time frame that you want to be there. If you want to ask, let's say 100 people, maybe you only have to stand there for an hour or so. But this is going to lead to biased or unrepresentative results. Of course, the people that walk past a library are going to be reading more books on average than the average person in the population that you're trying to generalize to, which is maybe all of the residents of a certain town or of a certain country. So bias, we learn, is when we systematically under or overestimate what we're trying to estimate based on the methodology that we use to gather the sample. Convenience sampling isn't the only bad sampling technique that we learn about. We also learn about voluntary response sampling, and that's where you cast a wide net, but you allow individuals in the population of interest to respond. And this could be a problem if the people that respond are different in some way systematically than the people who choose not to. So this is any time that you allow people to call in, text in, write in, scan a QR code, send an email with feedback. Again, you're going to systematically have people responding that share some similarities that are not shared by some people choosing not to reply. Okay, so to a researcher, it's very obvious that we should not allow convenience sampling, that we should not allow voluntary response sampling. We obviously want to do some method for random sampling. But even when we, the researcher, does everything in our control, we do everything right, you can still end up with some problems that lead to bias. And on the screen, we see 3 of them. So there's undercoverage, and that's when certain members of the population are harder to reach than others, so they end up being underrepresented in your sample. There's non-response, which is when some individuals choose not to respond even though they've been selected randomly. Again, this can be a problem if several in your initial sample choose to go this route and they share similarities. And then finally, response bias, which is sort of a big catch-all. It can include things as simple as lying or feeling intimidating and not giving the true answer to a question that's being asked to them. And just remember, bias is not a luck situation. Bias is systematic. So again, if you think about voluntary response bias, that is self-selection by respondents. If you think about convenience sampling, that could yield favoritism by the sampler. And if we want to do away with both, what we do instead is random sampling, which involves the use of some chance process to determine which members of a population are going to be included in your sample. Now, the first method of random sampling that we learn about in the course is simple random sampling, which has the acronym SRS. And again, a bunch of the other ones start with an S, but SRS is reserved for simple random sampling. And the defining characteristic of this sampling technique is that a group of size n has the same chance as any other group of size n in the population of being included in the simple random sample. College Board allows for 3 methods of random sampling that they consider to be valid simple random samples. The first is choosing numbers out of a hat. And a couple things to remember there is you want to write the numbers on equally sized slips of paper. And again, you're going to number all the individuals in your population and place those on evenly sized slips of paper. You are going to shuffle, mix around those slips of paper and pick them out one at a time, being specific about whether you're going to return them back into the hat or throw them away, discard them once they've been chosen. But the key here is you need to mix them around every time before you pick the next number. Okay, and then finally, like with any of the 3 sampling techniques that are considered SRSs, you need to connect those numbers back to the original individuals in the population that you assign those numbers to. You're selecting individuals, not numbers. Okay, so that's with a hat. You can do the same thing with technology, okay, so that's like using a random number generator or the randint function in your calculator, but again, you label all the individuals in the population from 1 through n, you use the random number generator to obtain that many numbers, the number of numbers that you're looking for, making sure to skip any duplicates if you are choosing without replacement. And at the end, you are going to tie back those numbers that you selected to the specific individuals in the population that you're going to select for your survey or for your experiment. And last but certainly not least, you can use your table of random digits. That's table D, which again, you've seen all year in print form, but you'll see on the AP exam digitally if it were to come up for a question. But the idea, again, you number all the individuals in the population from 1 to n, and you need to think, what is the least number of digits that I would need to represent every one of these individuals? And you're going to scan from left to right, starting on a random row of the table, that many digits at a time, making sure to identify which numbers come up, skipping any duplicates if you're selecting without replacement, and in the end, tying them back to the individuals in the population. Now, there are other sampling methods that are just as good, if not better, than simple random sampling when certain conditions are met. And the first one we're going to talk about is stratified random sampling. And in order to understand it, you have to understand that strata are just groups of individuals in a population that share some characteristic related to what it is that you're trying to measure. And instead of just randomly choosing a group of size n from the entire population, the way stratified random sampling works is you are going to select small, simple random samples from each strata and combine them into one big random sample. Now, this works really well when the individuals in each stratum are similar with respect to that characteristic which is related to what you're trying to measure, and different from the individuals in the other stratum. And one of the statistical benefits from stratified random sampling is that it gives you more precise estimates of what you're trying to measure when compared to simple random sampling. It lowers sampling variability. And this slide is just a reminder that the word precision and the word accuracy mean 2 very different things in statistical contexts. So precision is about how close estimates, repeated estimates of the same thing from the same population are to one another. And accuracy is a measure of how close your estimates are to the true value that you're trying to estimate. And so, of course, the goal is high accuracy, high precision, but this is just a good visual so that you keep those 2 concepts straight in your head. And just one more visual from the sampling sunflowers example from class. If you remember, we were looking at estimating the number of healthy sunflowers in each little plot. where a big sunflower field is divided into 10 rows and 10 columns, so you're left with 100 little plots. And we're trying to estimate the number of healthy sunflowers in one of those boxes. And what you can see is that repeated random samples of the same size from that population. The center is going to be the same whether you use simple random sampling on top or stratified random sampling on the bottom, but the estimates from stratified random sampling happen to be a lot more precise, so a lot closer together, and they're both centered around the true population average number of healthy sunflowers per plot, which looks to be somewhere between 102 and 103 in this case. Now, another sampling technique which often gets confused with stratified random sampling is cluster random sampling. And in this case, you're typically going to use clusters that are geographically convenient. So all of the households, for example, in a neighborhood can be considered clusters. But it's very, very important that if you're going to use cluster random sampling, that you remember that you want the individuals in each cluster to represent the entire population, to be almost like little mini populations. So it wouldn't be like the same individuals that would make for good stratum wouldn't make for individuals that make a good cluster. Again, in stratum, you want the individuals to be as similar as possible and as different from the individuals in other stratum, whereas in cluster random sampling, you want each cluster to represent all the different types of individuals in a population, and so all the clusters should look fairly similar to each other. And that's because in cluster random sampling, instead of choosing a couple from each, you're actually just going to choose one or two entire clusters as your random sample. And again, that's geographically very convenient because you're going to save yourself a lot of time and money instead of driving to all of these different faraway places to get your random sample. Now, the last method for random sampling that is acceptable in the AP exam is systematic random sampling. And the benefit of this method is specifically when you don't know the size of your population. So say you're interested, for example, in estimating the proportion of voters that vote for a particular candidate on election day. Well, you don't know how many voters there are going to be. So if you want 5% of the population to be sampled, you wouldn't know what 5% is because you don't know 5% of what number. And so in systematic random sampling, what you're going to do is if you want 5%, that's 1 in every 20. 1 over 20 is 5%. And so essentially what you do is you start at a random number between 1 and 20, let's say 7. So the 7th person that comes to the polling location to vote, you interview and ask them what candidate they voted for, and every 20th person after that. And you guarantee yourself that by the end of the day, you have asked exactly 5% of the people who they voted for, but you didn't need to know at the beginning of the day how many people were going to vote that day in order to find your random sample. So again, if you're looking for information about a population, you really have 5 options. The 5th option being the census. And again, that would be getting information from all the individuals in a population. It's very expensive. We're rarely going to go that route. Instead, we're going to go for one of the first 4, which are the random sampling techniques. The most simple being the SRS, the simple random sample, but you can do stratified cluster systematic random sampling when it would be advantageous. And again, we've already discussed that each of those has a different statistical or practical benefit. Now again, the entire point in taking a sample is to be able to use the results from the sample to be able to make some statement or some conclusion about the entire population. Well, this only works if the sample is representative of the population. So you have to keep in mind that if you are not using unbiased sampling techniques, then the information that you're going to gather about the sample is not going to be generalizable to the population. Even when you do everything correct, you use one of the appropriate methods for sampling, the non-biased methods for sampling, you are expecting there to be slight differences between what you see in the sample and what is true about the entire population. And that comes from a fact that we call sampling variability, which says that different random samples of the same size from the same population can yield slightly different results. Now the good news is that later on in the course we learn about what we call sampling distributions, which can help us predict some of those differences. But the better news for right now is that there's ways that we can control those differences. The easiest of which is just to increase the sample size. So larger samples tend to produce better estimates than smaller samples. So if we want more precise estimates from our population, we can just increase the sample size in order to achieve that. Now, the methodologies that we learn in this unit are very useful for when we're drawing a random sample to give, for example, a sample survey to, but they're also super useful in case we want to randomly assign experimental units to treatment groups. Now, just to recap those differences, we've got observational studies, which can be either retrospective or prospective. In this case, we are not comparing anything. We're just trying to learn something about everyone that we've sampled. In a retrospective observational study, we are looking at existing data from the moment that we sample our experimental units. So we randomly select some individuals and we look only at information from that moment into the past. Whereas a prospective observational study randomly selects some individuals in the present and follows them and collects data from them as they move into the future. And just remember that one easy way to remember the difference between an observational study and an experiment is that treatments, and that's treatments with an S plural, are not imposed on participants in an observational study, but they are imposed on participants in an experiment. So generally speaking, experiments are more involved and difficult to conduct than observational studies. They require a lot more planning, and the logistics of them are just a bit more complex. You have to be a lot more careful and keep track of more variables and things. Why do an experiment then? And it's because it has more statistical power, whereas an observational study allows you to see that there's a relationship or an association between things. A experiment is actually what will allow you to say that A causes B, or that there's a cause and effect relationship between variables. So just like the first half of the unit where we talked about sampling design and we had a lot of vocab there, we're going to learn some new vocab words that relate to experiments in the second half of the unit. Now, an experiment is a statistical study where we actually do something to objects. We call those objects experimental units, or subjects if they're human, and those things that we're actually doing to them, those are treatments, and we call them treatments whether or not the study is medical. And once we impose these treatments on experimental units, we're interested in measuring their response to those treatments. Now, because most experiments have human subjects, we are interested in eliminating what we call the placebo effect, which is the fact that some people are going to just respond positively to the thought of receiving a treatment. And for that, we have placebos that we give, and these are treatments that are otherwise identical to the active treatment. So they smell the same, they look the same, maybe they taste the same, but they don't have whatever that active ingredient is that we are trying to measure the response for. One of the things that makes planning an experiment a little bit complicated is that you want to control for all of the factors that are not the explanatory variables that you're purposely manipulating. And one of the risks is a confounding variable, and this happens when two variables are associated in such a way that their effects on a response variable can't be distinguished from each other. And so you want to control for all possible confounding variables so that the only differences between the treatment groups are the explanatory variables that you're purposely manipulating. Now, one of the things that we learned in this unit is that there are 4 requirements that you need to satisfy to have a well-designed experiment that allows for statistical inference. And one of those 4 requirements is called comparison. And what that requirement says is that in order for an experiment to lead to valid statistical inference, you've got to have at least 2 treatments or 1 treatment and a control group. So just to be very clear, not every experiment has to have a control group, but where you're only testing the effect of 1 treatment, you have to have a baseline for comparison, and that's the control group. And sometimes the control group is given a placebo, other times it's not given any treatment at all. But the idea is that you treat that group exactly like you do the other group. The only difference is the active ingredient or the active treatment that you're giving the group that's not the control group. And we do that so that if there's a difference between the groups, you can attribute that difference to the treatment itself. Now, because of the placebo effect that we discussed a little bit earlier, we can sometimes use blinding in order to improve the efficacy of our experiments. So in a double-blind experiment, neither the researcher nor the experimental unit or the subject knows whether they're getting the active treatment or the placebo. And in a single-blind experiment, it's either the researcher or more commonly the subject who doesn't know which treatment they're getting. Of course, double-blind where possible is better. That way, there's no risks of the experimenter accidentally treating the subjects differently depending on whether they're getting the treatment or the placebo. Now, we've already established that there have to be at least 2 treatment groups in an experiment, but for the same reason why we didn't want voluntary response sampling, and we also didn't want convenience sampling on the sampling half of this unit, we have to let random chance decide which experimental units go to which treatment groups. We don't want to let them self-select, and we don't want to select ourselves. And again, if we do this, the hope would be that we end up with treatment groups that are roughly equal, so that the main differences that we see between them are the treatments that we impose on them. Now that we've got these roughly equal groups as a result of the random assignment that we did of the experimental units to the treatment groups, we apply control. And what that means is we want to keep everything exactly the same. We want to treat all of the different experimental groups exactly in the same way, so that the only difference between them is those treatments that we impose. And that way, any differences that we see, we can attribute to the treatments and not to any of the other things that we may accidentally manipulate. We've seen countless times throughout the course where increasing the sample size is a great idea. In the context of experiments, we want to increase the number of experimental units that receive each treatment. So definitely at least 2 per treatment, the more the merrier. Replication means that in the context of experiments, not to repeat the experiment multiple times as it might in AP Psychology, for example. So just to recap, in order to design what we consider a well-designed experiment, you're looking for 4 requirements, 2 C's and 2 R's. The 2 C's are comparison and control. And again, control does not mean control group. It means keeping everything constant except that which you are purposely manipulating. And the 2 R's are random assignment and replication. Now, there are 2 main types of experimental designs that we learn in the course, the first of which is the completely randomized design. In this design, experimentals are assigned to treatments completely by chance, and this is a map of what that looks like. So they all start out together, we randomly assign some to one group, some to another, and this could happen for many, many groups. You don't just have to have 2. Each group receives a treatment, and then we compare the response across the groups. Now, like stratified random sampling can be thought of as the fancier, more statistically beneficial in terms of precision way of sampling, we have randomized block design as the fancier or more precise way of assigning experimental units to treatment groups. So again, blocks are groups of experimental units that are known before the experiment to be similar in some way related to that which you're measuring. And what you're going to do is you are going to randomly assign experimental units from each block. And again, you're going to form those blocks before the random assignment. And this is going to allow us to account for the variation in the response that is due to that blocking variable. And again, this is very, very useful from a statistical perspective in order to really avoid the counter argument that maybe more of a certain type of participant got assigned to the treatment than the other, and that's really why you saw a difference between the response, not the treatments themselves. And this is a map of what that looks like. So again, we're comparing keyboard A and keyboard B and its effect on typing speed. And maybe we just want to make sure that we evenly distribute the participants in this study, or the subjects in this study, that are already smartphone users. So what you do is you form a block of smartphone users and you form a block of non-smartphone users. From the 4 smartphone users, you randomly assign 2 to get the keyboard A, 2 to get keyboard B. You do the same for the non-smartphone users, 3 and 3, and then you compare typing speed across them. But this way, you control for the fact that you're not going to end up with all the people that have smartphones in one group and all the people that do not have smartphones in the other group. And sort of the extreme version of a randomized block design is a matched pairs design where the blocks are of size 2. And you'll see this very commonly with like identical twin studies, where the idea is prior to the experiment starting, they recruit a bunch of identical twins and they randomly assign one twin to one treatment and the other twin to the other treatment. You also see common matched pairs design studies where you recruit the same participant to take both treatments in a random order. And so you get a bunch of participants, they each serve as their own block. And what you're randomizing is whether they get the active treatment versus the placebo first or second. So if you've done everything correctly, you've designed an experiment that has the 2 C's, control and comparison, and the 2 R's, random assignment and replication, and you see a difference between the response from one group and the response from another group? Well, there's two reasons left for that to have happened. Reason A is that there was really no difference between the treatments and random assignment led to the differences that you're observing, or reason B, that it really was the treatments causing those differences. And the bigger the difference between the response that you see across the groups, the more unlikely that it's the former reason and the more likely that it's the latter reason. And when those responses are too unusually different to have happened from chance alone, we call that difference a statistically significant difference, and that's what we're looking for. Now, this is the last slide of the unit, and honestly, an incredible one to pause and make sure that you really understand all the details. There's a difference between random selection and random assignment and how it leads to statistical inference. When you randomly select individuals from a population, we allow the results of whatever it is that we're studying to be generalized to that population. When you randomly assign experimental units to treatment groups, that's what allows for statistical inference when it comes to cause and effect, when it comes to saying that A causes B. So please, please, please make sure, of course, the best experiments include both. You randomly select subjects from the population that you want to generalize to, and you randomly assign those subjects to treatment groups, but sometimes based on what you are studying, it's not possible to randomly select people from the population. You have to rely on volunteers, and in which case you have to understand that you are going to be able to make claims about cause and effect, but only to individuals like those that volunteered for the study, not for all individuals in the population. That concludes our review of Unit 3. I highly encourage you before the mini assessment on this unit to look at some of these additional resources. There's Khan Academy, AP Classroom, we've got the Google Drive, which by the way, I've added in the common FRQ topics for each unit, so make sure to check them out, and also the actual old FRQs for this unit, which you can find on Stats Medic. I hope you guys have a great night, and I'll see you soon.